Hello everyone, my name is Yingguo. Thanks for the World Journal of Men's Health give us this chance to introduce our recent study. Title of the paper is A Simple and Non-Enzymatic Method to Isolate Human Corpus Endothelial Cells and Pericytes for the Study of the Erectile Dysfunction. The penis has a specialized vascular bed, and penile erection requires well-coordinated interaction between the vascular endothelial cells, smooth muscle cells, pericytes, and neuronal cells. The vascular endothelium plays important roles in the regulation of blood flow. Parasites are known to play critical roles in the vascular development and homeostasis. Therefore, the purpose of this study is to establish a simple and non-enzymatic technique to isolate endothelial cells and pericytes from the human corpus tissues and to establish an in vitro model of erectile dysfunction for the study of high glucose-induced angiopathy. For primary culture of the human conus endothelial cell, human conus tissues were implanted into the material gel with vascular endothelial cell growth factors in culture dishes. For primary human conus parasite culture, conus tissues were settled by gravity into the cell culture dishes. We performed the immunocytochemistry to determine the phenotype and the morphologic changes from the passage 1 to passage 5. To establish an in vitro model to mimic the diabetic ED, the primary cultured cells were exposed to a normal glucose and high glucose conditions. We also performed the tube formation to evaluate the ability of the conus EC and PC. In the figure 1, we can get positive immunostaining with endothelial cells and parasite markers in human conus tissues. It means the EC and PC exist in human corpus conus tissues. Next, after isolate the EC and PC from the human corpus conus tissue, we also staining the primary cultured cells with different cell markers. Primary cultured EC showed positive staining for EC markers BWF, but not the parasite marker NG2. In contrast, human corpus conus parasites showed the positive parasite markers NG2, but not the EC marker VWF. We also found the morphologic and phenotype of primary cultured cells were changed according to the passages. Both two kinds of cells show positive cell markers. However, from passage 4, these markers' expression is decreased significantly. This data suggests that the primary cultured EC and PC is better to be used from passage 1 to passage 3. So next we want to know what kind of cells it will be changed from passage 4. We found the morphology and the phenotype of primary cultured cells were elongated in size at passage 5, which is similar with the fibroblast. And the immunostaining result also showed positive staining with fibroblast marker FSP1 at passage 5. So from these data, the primary cultured cells will be differentiated into fibroblasts in high passages. Finally, to test the ability of the primary cultured EC and PC, we performed the tube formation assay. We found the primary cultured EC and PC can form well-organized capillary-like structures in normal glucose conditions. However, the number of the tube was significantly decreased in high glucose conditions. In summary, we established a simple and non-enzymatic method to isolate human conus EC and PC, and an in vitro model for diabetic ED. This will be a valuable tool for evaluating the candidate therapeutic targeting angiogenesis for diabetic ED. Thanks for your listening.